All right. So first of all, I'd like to thank Dylan Davis and Davis Dylan for reaching out to me and giving me this wonderful opportunity to meet all of you for the Life After Graduate School meeting series. And um, I'm really grateful to talk about um, my personal experience and insight into the biomedical data science. And I did my PhD under the supervision of Dr. Muttalib with my dissertation on gender matrix model in 2010. And after my graduation, I worked in a various broad biomedical field of biomedical data science at various different institutions. And as I just introduced, last year I found a physics executive position at Drake University in Des Moines, Iowa. And I had an opportunity to teach undergraduate mathematics and physics courses. And before joining the Drake University, I did my postdoctoral training in biostatistics at Khan School of Medicine in Mount Sinai in New York City, where I had an opportunity to apply biostatistical analysis method to the problem of understanding human health and disease. And I'm excited to actually give you some sense of what's going on with this new emerging field. It has been actually there for a long time, but from my perspective, what's going on in this field with the ultimate goal of promoting human health and treating human diseases and preventing diseases. So please feel free to interrupt me anytime if you have a question. So to begin it, I'd like to introduce you briefly about biostatistics. So biostatistics is a field of research focused on developing or applying statistical mathematical methods to biological science problems. And one of the active research area where biostatistics plays a crucial role is analyzing clinical data to study drug effects. So in this area, researchers aim to determine whether a particular drug is effective for treating patients. And another active research area is investigating cellular molecular mechanisms that underlie human health and diseases using omics data. And omics data refers to the data obtained from entire set of biomolecules from the cell. For example, the genomic data refers to the entire DNA sequences obtained from cell, and then the transcriptome data might hear is from the transcriptome data refers to the data obtained from expressed RNA molecules cell, so entire sets. And my personal experience is in the second research activity, mostly, but I found that the ideas and techniques can be applicable in different research area. And these two, it's not just two, but the research activity that I'm mostly acquainted with, these research activity deals on the ideas and techniques of statistics. So I'm going to talk a little bit about what I have begun to realize, like what the statistics are. So statistics is a science of collecting and summarizing and exploring data, and also drawing conclusions and making decisions from data. And there are two different branch of the statistics. One is called descriptive statistics. This involves summarizing and exploring data. For example, drawing histogram or drawing scatter plot for these examples. And then and drawing conclusions or making decisions from the data involved inferential statistics. And the inferential statistics involves, for example, conducting statistical tests using this score or estimating confidence intervals. And both of these activities are the descriptive statistics and the inferential statistics play this important role in biomedical data analysis. And I think this is really exciting time to learn about statistics and biostatistics um, because there's so much potential to understand how the biosystem functions at the level of molecular cellular resolution using omics data. And this all became possible due to Human Genome Project and ensuing projects and also advances in omics and science technologies. And in these efforts, these research efforts have enabled systematic investigation of basic building blocks of human genome and also the genetic differences among human individuals. The Human Genome Project is a landmark global scientific efforts launched with the goal to determine first DNA sequence of human genome. And during the which is carried out between 1990 and 2003, and they determined the DNA sequences of several organisms, including human genome. And the organism's genome refers to the complete set of DNA instructions which is found in every cell of an organism. And the DNA is, as you know, is made of four different chemicals or the nucleotide, which are represented as A, C, and G, and T, the four letters. And the ordering of this letter, the DNA sequence, encodes information how the cells in the organism operates, what to do, when to do, and where to do, and what to do. Okay. And the DNA sequence is tagged into structure called chromosome. And in human, the human chromosome consists of human genome consists of 23 chromosomes, and we all inherit one copy from mother and one copy from father, and each of these copies contains roughly about 3 billion DNA sequences of letter A and C and E and G. And it turns out that our individual human genomes are far more similar to each other than they are different. So it's frequently stated that any two-person genome is more than 99% identical to each other. And the differences in individual genomes are called as genetic variants. And the predominant forms of the genetic variants are known as single nucleotide variants, which is illustrated as a single letter change in DNA sequence between individuals. And these differences are presumed to contribute individuals different physical characteristics and also health conditions or how one responds to a particular medication. And so these discoveries about a human genome led to a large number of collaborative research projects aimed at determining genetic variants at the single base resolution, which differs between healthy individual and individual with certain diseases. The hope was that knowing the genetic variants, which is more frequently found in people with disease, could tell us how to prevent and treat human diseases. And so in these studies, typically the researcher will recruit healthy individuals called cases, and then individuals with certain diseases called cases. And then they will sequence individual genomes and then analyze whether there are genetic variants whose frequencies differ between these two groups. And if so, then it would indicate that the particular change in the genome is associated with the particular disease. And let's imagine that, hypothetical genome-wide association study, the researcher recruited 500 healthy individuals for controls, and then 500 individuals with some certain disease cases. And from the sequence genome of this thousand individual, they discovered that there is genetic variants somewhere in the genome, whose frequencies in control where in the control sample the letter is often the letter A is often changed to letter A in the people with certain disease. So they counted how many times A and T occurs in these two groups. And then they would pose a question whether having an A instead of T is associated the risk of having this disease, particular disease. So to address this question, you can turn this problem into a statistics problem. To turn this into statistics problem, we ask whether the proportion of A in the case the individual disease differs from the proportion of A in control individuals, the healthy individuals, and and then we then come up with a model to in this model we will model the we consider the probability of success. So the probability of model the proportion of A observing an A as a probability of the success in a binomial distribution. Like the coin toss. Consider observing an A as head and observing T as a tail, or more genetically, you can consider having an A as success and T as a failure. So we consider in this model, in each observing an A or T as an outcome of a Bernoulli trial. And we make an assumption that the probability of observing A is the same for all trial or all individuals in cases in control. And then we also make another assumption that each trial is independent, meaning that observing an A in one individual is the same as observing is not affected by observing an A in another individual. And applying this model to this problem, you need to consider that the proportion of A in cases and controls are unknown to us. So we are going to determine whether the proportion of A in cases and control could have been the same. To determine this, we need to test whether the proportion of A in cases and control, although there's apparent difference in the proportion, whether this difference actually came from just random chance alone. So, for example, if you look at the proportion of A in case it's 200 out of 500. So this is roughly about 40%. And in control, the proportion of A is 100 out of 500 individual. So it's 20%. There is apparent differences, but we need to test whether this apparent difference is big enough to say that there is a true difference between these two groups, rather than it just came from random chance. Because if you sample A from large population, the proportion A in one sample can be different from another sample. And to do that, to determine this, we'll construct a test statistics, which represent the chance distribution that reflects how likely these two proportions came from the same underlying distribution. And this test statistic numerator represents the estimated difference in the proportion of A in case and control. And then denominator represents the spread in the proportion, assuming that they came from the same underlying binomial distribution. So if the proportion of A in cases and control were the same, then the tested statistic would approximately distribute as normally with mean 0 and the variance of the standard deviation of 1. And with 95% of the chance, the test statistics will be between negative 1.96 and positive 1.96. And having calculated the test statistics from the data that we have, which is 13.3 in this case, much greater than 1.96, indicates that it's very unlikely that the two proportion in the A in the case and controls are the same. And it's very likely that the proportion of A in case is greater than the proportion of A in control, meaning that having an A is associated having the particular disease under consideration. So a number of researchers around the world applied this genome-wide associated study in many different diseases for the last couple of decades. And this is the snapshot of the result, which is the genetic variance across 23 human chromosomes. And that's color coded by human organ system or certain disease category. And you can notice that the dispersion of the genetic variance, the colors across 23 human chromosomes for any disease category or organ system category. And researchers have realized that in general, it's difficult to translate what they found from the genome-wide association study result, which is knowing which genetic variants are associated with particular disease into the molecular cellular mechanisms that leads to particular diseases. And this difficulty arises in part due to the fact that for many human diseases, the pattern of inheritance is complex, that it's been really challenging for researchers to make sense of how these genetic variants altogether contribute the function of relevant organ system, and that eventually leads to particular disease. And so to better comprehend how these genetic differences contributes human disease or health, the researchers embraced a systems level approach. The fundamental concept of the systems level approach is thinking that the human disease and the traits are outcome of interactions of basic building blocks of the human body. For example, the cells, the functioning of cell is result of the interactions of its molecular component, genes and other biomolecules derived from the genes, and also the functioning of tissue or organ system is an outcome of collective interactions of the cells and so forth. And so therefore, to understand how the genetic differences contribute to human health and disease, it is necessary to combine the genetic informations with the molecular interactions or cellular interactions, and then linking to the observed phenotype of the diseases or health. And the omics data has been valuable in this regard because it offers the quantitative and also qualitative measurements of the various biomolecules in a cell. And these information can be used to study how individuals with certain disease and healthy individuals are different from each other. And to study these, we need to develop accompanying statistical and computational method to discover what are the cellular or molecular interaction profile that's responsible for human health and disease. And recent advances in omics science and technology provides an exciting opportunity in this research direction. And there's tremendous and flourishing efforts toward deeper characterization and quantification of pools of biomolecules that leads to the structure and function and dynamics of an organism. And these efforts allow us to study great details of the activities of biomolecules. For example, how the DNAs are, how much DNAs are transcribed into RNAs and how much of the mRNAs translated to proteins at very particular biological context. For example, it's known that building human central nervous system is a complex dynamical process that takes place a long duration of time. And previous analysis has shown that the transcriptome, the entire set of expressed RNA molecules in brain cells undergoes profound changes during human brain development. And it's also known that human brain disorders have discrete ages of onset and progression. And this presumably arises due to genetic and also environmental perturbation at various developmental time points. And these can lead to differences in omics profile in people with diseases and healthy individuals. And more recently, the omics science and technology allows to characterize or quantify the spatially resolved single cell resolution using optical imaging technology. With these technologies, we can gain an insights about what's going on with thousands of molecules inside of thousands of cells at very particular location of the brain tissues. So thinking of the progression of these omics science and technology, it provides so much opportunities, and that's really exciting. But at the same time, it poses great challenges for researchers, how to integrate all these expanding informations to properly learn about how the biology functions at cellular molecular resolution. And I think that thinking about developing or applying biostatistical analysis methods for these omics data, I think the ideas and techniques used in the regression analysis is interesting to understand. So in the regression analysis, one seeks to identify or quantify approximate relationship between variables. or how one variable depends on another variable, which we call as response and predictor. And you can apply the regression analysis idea into the problem of the genome-wide association study. For example, in that case, we would consider one's health outcome, whether one is healthy or one is diseases, can be considered as a response, and the number of the genetic variants at particular position as a predictor. And given that response and predictor data, we can fit a straight line, a regression line, and then see if this regression line slope is non-zero. If the slope is non-zero, that would indicate that the two variables are correlated or associated. And to do that, we need to apply appropriate test statistics for the estimated parameter, slope parameter, and also its sampling distribution. There's an advantage of doing the genome-wide association study using the regression analysis approach because it's much more flexible, and it also is applicable to various other biostatistical problems. And I'm going to illustrate how actually this regression analysis techniques and idea can be useful to studying the interactions between molecules. So for this, I'm going to assume that we are interested in studying the quantity activity of one molecule, which can change due to directly or indirectly interacting other molecules. So in this case, this model may be relevant to a situation where the promoter of the gene, which is actually which can reflect the activity of a particular gene, is controlled by another molecule called transcription factor. And yet another, this transcription factor is yet controlled by another molecule, protein complex. So in this case, we can express the quantity of different molecules as a set of a regression equation. For example, the theta represent correlations and epsilon to represent the noise. And then you can rewrite these equations into a matrix form. And then with the help of the matrix algebra, we can easily show that the covariance matrix tells us about what's going on with the molecular interactions in these three molecules. And more in particular, the non-zero element, off-diagonal element, will tell us non-zero element of the off-diagonal element of the inverse covariance matrix tells us about which genes are interacting, which molecules are interacting. And the problem in the statistics is how to actually learn this mathematical relationship from data. And to learn that, we need appropriate test statistics for these model parameters. What is the estimated what the estimate and what the sampling distribution of the test statistics, the model parameter. And here I'm trying to sort of summarize what I think as an opportunities and challenges. I think that you'll probably soon realize that there are much more actually set of opportunities and challenges. And I'm just trying to give you a sense of what I think is already available and what I think is interesting to tackle, but there's so much activities going on. It's not possible for me to even try to give you all entire activities going on in the biomedical data science. And so the regarding the opportunities, there's already tons of omics data publicly available out there, which provides really accurate and comprehensive measurements on various different biological systems. It's already out there, so if you are interested, you can use it to do your research. And then the second thing is there's a good motivation and interest in understanding the molecular cellular mechanisms underlying human diseases. This will help to improve promote human health, and then this will help to prevent and treat human disease. And in that regard, there's so much interest and good motivations. And regarding the challenge, uh, as I just mentioned, the omics data, we try to quantify, characterize its entire activity of biomolecules in the cell. So if we consider the human gene, the number of the human gene is more than 20,000. And how these 20,000 genes actually can contribute to particular health outcome is not easy to actually tackle. What what is actually the model that's underlying this? So that is one really interesting um, active research area. And then another active research area is there's so much rapid progression with omics data science and technology. And anytime new technology or new science comes out, there's a new data comes out, and people don't know exactly how actually to incorporate these expanding data into existing with existing data. So there's always need for developing new methods, mathematical, computational, statistical, and that's also challenging. And here I'm trying to sort of summarizing that I'm aware of as an opportunity, perhaps for you after graduation or right now. There's so much to actually homework for you to do, probably. So I'm just trying to give a sense of what's out there, and then I'll also give you some, I guess, how do I say, I will share my personal experience or my thoughts in this. So both funding agency, the National Science Foundation or NIH, National Institute of Health, has a lot of opportunities. In terms of opportunities, I mean, yeah, I don't even know it. Yeah, for example, like the NIH has 27 institutes, and all these different institute has fellowship opportunities and grant opportunities. And it's hard to actually navigate. But one, I guess, if I'm thinking about the scenario where after you graduate, mm, I think this discussion came up slightly, but usually within after graduation of like 3 to 4 or 5 years, some of the opportunities actually are not going to be eligible. So, for instance, the next fellowship that's probably only available until you stay um, 3 to 4 years after you finish PhD. And then it's pathway to independence award. If you're particularly pursuing academic career, if you want to be a faculty member in a research institute, this is one of the really exciting opportunity because this funding allows you to actually develop into a faculty member, not only just getting a training, but that funding clock ends at 4 or 5 years after you finish your PhD. So one thing that I would like to actually recommend, although I wasn't successful, I wasn't able to get the grant. Keep in mind that if you plan ahead and then if you can actually diligently figure out what would work for you, and if you can get a fellowship or sort of grant, then your chance being successful as a scientist increases. So that's one thing that I realized because after I've gone through several postdocs, it became challenging after like several years. No more these opportunities are available. But it doesn't mean that there's no opportunity. It can happen like to anybody, right? So it's not guaranteed that we all actually like jump back to the next stage. It takes time. You have to explore and you have to learn. There's so many things to do. So time-wise, it's really difficult to control. But be aware if you want to be a successful and if you're looking for academic career, then you really have to think ahead and then try to set the steps properly to get to this level as soon as you can, as early as possible. So in that way, your chance to be successful increases. But at the same time, yeah, don't worry. Even if you pass that point, there's still chance. I think in terms of the opportunity, it's always there. That's what I feel. But eventually, what I realized recently is this. So the typical challenge is figuring out your own research agenda, something that you personally want to pursue, like research agenda or whatever that is. And once you realize that, then I guess question is how can you actually contribute with your own idea? I'm talking about the biomedical science and the problems, but they all apply the same way, right? Other places. How I can contribute with my own knowledge and my own skills and my passions, whatever that is, to the particular problem or community or and we have to have sufficient I guess training to do that. So I think so I was yeah and so I was preparing so the thing that I realized after like almost decades after graduation is it's not like opportunity comes to me. It's rather I create a path and then I try to sort of figure out what opportunities are relevant for me. And then this comes to you. It's not like, oh, there's opportunity. So I can just sit down and then it just comes to me and I can just take it. It's not, and maybe I was like that in the beginning, right after the graduation. But then over the course of time, what I realized is that, yeah, if you have a will to do something and if you go, you take the steps properly, then opportunity actually comes into you. I'm still actually in the middle of doing something that I think I'm sort of trying to do, but yeah, that's how I feel. And also, I remember this was actually one of uh, the reception party where the graduate students actually finish their PhD, and and I was actually sitting there, and then there was someone actually who left me earlier, and the person said that the hard work pays off. I think that's the truth. Hard work always pays off. Other than that, I think what I realized is it would help if you plan ahead, if you think about what is the exciting problem to you, and if that aligns with your own passions, then you will see more opportunities coming into you. Yeah. So that's I think something that I was thinking about, and I would like to thank the UF Physics community. It's an excellent community. I love it. And also, I'd like to take this opportunity. I'd like to thank my PhD committee members. I asked them so many times the recommendation letter. I had to bother so much. And then recently I was really reluctant because I bothered them so much. So I tried to apply places where they ask recommendation letter. I'm not writing. Once I get past the first test, I just ask them before. So a lot of times it happens that you have to send a recommendation letter to just apply for the position. And then I don't know, I don't remember. I applied more than 100, at least at the minimum over the course of time, more than, maybe, maybe not just 1,000, but quite a lot. And every time, I mean, it takes time, right? Writing. But then I felt like, I don't want to do that anymore. So I just want to see if there's a real opportunity. Then I'll just ask them. I just bother them to write a letter. But it happens that actually right after the graduation to get a fellowship, it always asks you probably the recommendation letter. And so I bother them so much, and they never complained. They were always willing to write a letter. So I mean, I have to appreciate all their efforts. And, and thank you so much for everyone showing up and kindly listening to my talk. And I really appreciate all your help. Thank you.
We can transition to a more open discussion. I have a lot of questions right now.
Sure.
Does anyone have any questions that they have that they want to ask? Yeah, sure.
So, what made you shift back to teaching?
Uh, it's something actually coming back. Um, I guess in teaching position, you have an opportunity to actually merge both aspects, education and the research. Definitely, if you go to a research position, it there's more time to actually do the research alone, right? But that's when I was postdoc. In postdoc, it's so if you write your own grant, if you're on your own grant, then even the postdoc papers their own actually research problem. But from my experience, I was always on grant, someone else's grant, and that means I'm doing a lot of work, I'm learning a lot, but at the same time, uh, at the end of the research, I didn't think exactly this is the direction that I was pursuing for the future. I mean, there's so much to learn, so it's okay to actually being getting a training somewhere many times, but that's fine. But to pursue your own research agenda, your career, you have to find an independent position. That's where I started thinking hard, and even if it's so, I guess it depends really on what institution you go. There are some research focused institute, and then there's more education focused institution. But even if it is small, there's an element where you can actually pursue your own research. And also, I find that after teaching one semester, I realized that teaching is really helping me a lot because you have to think really simple. You have to make it easier. And I haven't thought about. Sometimes I thought I knew, but then I realized I don't know it exactly. And that actually, while teaching it, it actually helps me to actually think about the problem more carefully. So right now, to me, I think it's okay whether this is more teaching focused or research focused. You can just develop yourself. But yeah, um, to your question, yeah, on the surface, it looks like I'm coming back, but on what happens is this sort of progression in me. It's okay to be anywhere. I can I can do whatever I want given the opportunity. So that's I think where I'm sort of.
I have a question about, have you ever had any inclination of going towards industry, or are you always
There's always opportunity. There's always opportunity, especially regarding the disease mechanism. That's what actually industry is looking for. Because if you know the mechanism, then you can actually develop drug,
yeah,
right? So there's tons of biomedical, pharmaceutical companies that in, like with specific interest in like finding the drug targets. I mean, it's difficult to actually count. You just Google, and there's so many opportunities out there if you want to go that direction. And also industry has post opportunities. You get to train in certain area, and then you go into that direction. Uh, for me, I don't know. So I think it's either way is okay as long as you can contribute with your passions or your if the thing is actually in your own, I guess, pursue, what you pursue, right? It doesn't have to be academia or it doesn't have to be industry. It doesn't really actually matter. What actually suits you is actually the thing that you have to figure out. I don't know. I haven't tried the industry. It might work better than what I'm doing now, but that is open question. But what I think is important is somewhat now I'm seeing a different way is it's me actually who creating the path. And as I'm creating the path, maybe I can work in like academia or industry, and just following the steps, not like deciding whether I should go industry or whether I should go to academia. It's not that decision for me. It's more of whether I am making progress in my own way. If I make progress, if it leads to industry or if it leads to academia, that's actually, yeah, I guess how I would go about. Yeah, I thought about it, and also, I mean, as far as exploring, I, not all my actually experience using entirely academia. I mean, it was in a academia setting, but there was some problems, and the work that I did was more in the industry side. So, I mean, in terms of exploring, can be open, and you can try and see if it works. And there's also people who are who worked in the industry and comes to the academia because they gain actually insights about what's really necessary in academia, and then they come with that idea. So you can switch back and forth. So I don't see that there's a like boundary. And if you go on one side, then you can come back, or you can, yeah, there's always opportunities, and which opportunity suit to you probably depends on what stage you are in, or yeah, yeah.
Uh, having having developed your career into US, would you be able to comment a bit about opportunities abroad? So, for example,
Mhm.
why one to find maybe an academy position in another country? What do you think the opportunities are?
In outside of US.
Yeah.
So I think that's interesting question. I don't know exactly if I can answer your question properly, but one of the places that I visited here is Perimeter Institute. It's really prestigious theoretical physics institute in Canada. So this is one place that I learned from my advisor, Muttalib, as I graduate. He recommended to look at this place, and I think they do really cool stuffs. And so, I mean, I don't think in terms of looking for the opportunity, you should limit yourself. Mhm. It's everywhere, and what just figuring out what suits to you is actually good, and then you can connect people there. Then the opportunity will open up. So I don't think it has to be limited to US. So one thing that I realized, I actually right after the graduation, I went to uh, an institute in Italy. I was there as a visiting scientist for 1 month after my right after the graduation. Actually, before the graduation, actually before graduation ceremony, I left there. I was there for a month because my research work was related to a person working there. And one thing that I realized as I go to Italy, I didn't know how to speak Italy. I was much more comfortable to speak English than like other languages. That actually somewhat practically limited myself considering more job opportunity considering in the US than outside, because then I also have to think about learning the new language. Although the institute itself actually encourages to speak English. Um, yeah, yeah. In terms of the opportunities, I don't think you have to limit yourself. It can be anywhere. Um, yeah, it's just figuring out what suits you to at that position at that point, and whether that's in your direction. And that's I think what really matters. But yeah, hope I answered at least a little bit of what you're wondering about.
Uh, Do you think like doing a postdoc fellowship for like independent research is like required for like these faculty jobs that are research focused, which like they specifically look at the candidates to see if they have done uh, fellowship related postdoc, not like under a lab.
Uh, I don't know exactly. I understood your question. So, to be successful in academia, I think it's important to have your own, I guess, research agenda, right? And to develop that, I think the fellowship is better in terms of because when you write a fellowship, at least part of this, although there's a training element, whoever hosts you, there is an element that you are actually writing, and that actually helps you develop. But when you go to postdoc jobs, which offers you a stipend with the host PI's idea, then you have to learn the PI's idea first, and then you have to develop your own. So it takes, I guess, more time to figure out. But it can be that the PI actually has very good idea. He already has actually is already far out there, and you can learn from him, and then you can actually develop yourself from there. Then actually that's also a good opportunity, right? So it's really difficult to say only one like way. If this is good and that's bad. It's more of just figuring out. If I'm really at the beginning, then I have to find a place where I can really learn more rather than really trying to be far out there, right? Then if you're in a position where this is too difficult for you to actually do, then you may not learn too much very well. So I guess the focus is over the training. Am I getting trained? Am I making progress in the direction that I'm trying to pursue? So in any case, I think, yeah. So thinking again about the direction, if you can write a grant, that helps you to think about what you really want to pursue, right? So even if it fails, you have tried. So I think in that sense, that I think always good direction to pursue. But even if it's not like happening, you can still pursue. Like learn from others, and then also build your own agenda, and then figure out. So opportunities sometimes comes not exactly what you want, but you can make it happen with your own effort, even if in the beginning it's not exactly the way that you want.